Here we are at the connective tissues, so let's get into it. Going through, I see we could talk about the major functions of connective tissues, but there are so many different connective tissues, they have a lot of different functions. So we would learn the functions of each distinct tissue, not them as a group. And these were the some of the major characteristics. The extracellular matrix is a big one, and being derived from mesenchyme is the other big one. And these are the cells that you would need to know. Let's see, fibers and ground, blah, blah, blah. Where are we? So there are some slides. These are from your slides. And these are some slides on ground substance. It shows sort of how to picture proteoglycans and what? Fibers. I talked about fibers, collagen, elastic, and reticular. And one thing you'll notice is they have to use different stains to make them visible. So reticular fibers aren't easy to see. So they will sometimes stain them with silver, I believe, instead of the normal organic dyes they use. And elastic fiber is the same thing. So if you can see elastic fibers, chances are everything else is a different color and you're seeing different objects than you're used to. The typical, this is the typical H&E stain that shows proteins very well. So collagen would show up very nicely. Actually, this is not H&E. Where's some regular H&E stain? These slides are trying to make me crazy. That's what's going on. They're trying to make me crazy. I'm kidding. This is H&E stain. H&E stain shows purple nuclei and kind of pink protein. So something like dense irregular connective tissue with random jumble of thick collagen fibers would have a lot of pigment. It would be mostly pink with purple nuclei. So, but I got ahead of myself. We were looking at the fibers. So then we go into specific connective tissues. And with connective tissues, I separate them into classes of connective tissues. So I would say connective tissue proper, I guess, is a class, and the two subclasses are loose and dense connective tissue for connective tissue proper. Let me back up. So some terminology that would help make this consistent is I think of various classes within connective tissue. And those classes are connective tissue proper, cartilage, bone, and blood. Within them, some of them have subclasses, like cartilage has hyaline fibrocartilage and elastic cartilage. Connective tissue proper has two subclasses and subclasses within them. The subclasses are loose and dense, and the subclasses are areolar, adipose, reticular, regular, irregular, and elastic. We'll get to all of that, but just be aware, you'll see me say like subclass. Subclass is going to be within one of these four classes of connective tissue. And let's see. So we start with the loose connective tissue proper, and this is areolar connective tissue. This is kind of fun. This drawing shows transitional epithelium with a lamina propria deep to it, or basal to it, and that would be made of areolar tissue. So let's see what we can see in this areolar tissue. I see nuclei, so cells that are separated from each other, and they don't appear to be associated with any of the other big structures I'm looking at in any obvious way. So I would expect those to be fibrocytes or fibroblasts, so cells from CT proper. And I see, let's see, thick bundles, which are probably collagen. And then these thin fibers might be reticular fibers or elastic fibers. Areolar connective tissue has all the kinds of fibers, so you might find elastic or reticular. Whoever took this picture stained it in a way to make them visible, and so it might be you can't normally see the elastic and reticular fibers. But there's a lot of space in this tissue. There's a lot of space, and again, its function. Anybody remember the function? Why don't you pause the video and tell me what function I talked about in class? I said it has space for cells to move through. It has space for fluids to move through. It just is supposed to provide space for the body to get access to epithelia. Here's another picture. Sometimes it's very ugly. But again, what can we see? Cells, cells with nuclei where we can't see the cell, and then thick bundles of collagen, thick bundles and wavy collagen, and these thin fibers, which are either elastic or reticular. So it's really the space. The space and the jumble of different types of fibers are what make this clearly areolar. And a drawing of all the things going on in that tissue would kind of help you see that you could expect it to look like that. Okay, next is adipose connective tissue, and it has a distinctive appearance where nothing stains very well except the nuclei. And so what you see looks like these empty spaces, and each one is a fat cell. And what you're seeing is a vacuole containing a giant droplet of fat. And these cells can convert that to help, well, they store that so that we can convert that to energy. Let's see. Reticular tissue, again, I told you it forms a kind of three-dimensional scaffold for lymphocytes to sit on, and here you are looking at a bunch of lymphocytes sitting on reticular tissues. And what you should envision is fluid flowing into your screen past all these things, and all these cells are sensing what's in the fluid. And by sensing, I mean they have cell surface receptors that are trying to bind specific molecules in the fluid. So areolar, adipose, and reticular connective tissue proper are the three loose connective tissue proper. So now we look at the dense CT proper. That includes dense irregular, which has this kind of jumble of collagen. So the most dominant thing, the most dominant structure is these big pink structures. And I just see them, I see waves, and it's hard to tell where they're going. So dense irregular connective tissue makes up a lot of the dermis of the skin, and also fibrous capsules of joints. It holds them together. Dense regular is easier to see. It looks organized. Even though these are wavy, they are parallel to each other. And there are cells, the fibroblasts or fibrocytes are in between the collagen fibers, so they're kind of excluded by the collagen fibers. So this would be in a tendon or a ligament. And then elastic connective tissue is really distinctive because usually when we see it, it's relaxed and contracted, but it can be stretched and it will spring back. So yeah, walls of arteries, and I'm not sure off the top of my head where in the vertebral column we would see elastic connective tissue, but definitely arteries. And here's some histology. This is awesome. Dense irregular connective tissue. This is what the dermis looks like, where you have these random oriented collagen. So some of these collagen bundles are on the plane of the slide, and some of them are going into the slide, like perpendicular to it. Yeah, this is pretty zoomed out. Here you're seeing blood vessels with cells around them, and then elastic connective tissue proper with a special stain to make the elastic fibers red. And if this were black and white, we would still be able to tell what it is because these elastic fibers are distinctive looking. Let's move on to cartilage. Cartilage has three subclasses: hyaline fibrocartilage and elastic cartilage. Cartilage has chondroblasts and chondrocytes, and the chondrocytes are what we will find in the lacunae. So let's look at hyaline cartilage. The first kind of cartilage is hyaline cartilage, which has lacunae with chondrocytes in them, and those would have nuclei. That's how you can tell what you're looking at. And this featureless extracellular matrix, which is highly viscous and springy and avoids compression. So unlike collagen fibers, you could use hyaline cartilage when you need to resist compression. This is trachea with a lumen here. That's where the air would be in the trachea. And this would be, pseudostratified ciliated columnar epithelium. A whole lot of other stuff, including areolar tissue. And then cartilage, hyaline cartilage. And when you magnify it, you just see that the lacunae with kind of featureless, they're like floating in this featureless extracellular matrix, and that's what they are. That's hyaline cartilage. The other two types of cartilage have fibers in the matrix, and so they don't look like that. So one is elastic cartilage, which does have kind of a distinctive shape to the lacunae, and they generally, I do see them, there are more lacunae and they're closer together, and you might see the elastic fibers. And it's found in two parts of the body that start with E, so the ear and the epiglottis. That's elastic cartilage. Fibrocartilage would have more extracellular fiber, more fibers in the extracellular matrix compared to lacunae. And it's going to be in places where its tensile strength can resist shocks. So in the intervertebral discs is one of the locations where you'd find fibrocartilage. So in different colors, which is it's always good to look in different colors because you don't know which type of stain will be in the picture I show you. Where do we see a featureless extracellular matrix? Up here, that's got to be hyaline cartilage. And then elastic cartilage is going to have a lot of lacunae, basically. Very close together with fibers, clear fibers in between. And then fibrocartilage will have more space between the lacunae with, again, a lot of fibers. So that's cartilage. For bone, we have compact, well, let's look here. This is a cross-section through a femur where we have basically compact bone on the outside and spongy bone on the inside, and in the shaft of the bone, it's actually hollow with a space for yellow or fatty marrow, and the red marrow where blood cells are made, would be floating in between the little bone bits in this spongy bone. So I hope you can see why they call it spongy bone. It's got spaces that are connected by structures. Well, the structures are bone, so it is rigid. And you'll see later, the lines kind of follow stress lines where you'd expect the stresses to be in this bone. So they actually make it a little bit stronger, whereas the compact bone is by far the strongest part of this bone. So let's get back to it. Compact bone, a cross-section of it is what looks kind of like tree rings, but what you're seeing is much more awesome than tree rings because there are cells everywhere in this compact bone, in this super dense bone with no visible features. There are cells everywhere. They're just too small to see. And if you follow, I don't know if you can see how well you see my cursor, but the lines that radiate out from the center in bone are actually holes, and the cells have projections that fill the holes. And what they do is they touch each other and they can communicate. And also that has huge surface area for breaking down or making bone. At the center of one of these Haversian systems are blood vessels and nerves. And so it's a bunch of these connected to each other that make up compact bone, make it very strong. So spongy bone is going to look very different from compact bone. Yeah. And we will look at this in detail in Unit 3. We'll look at the parts of bone and what all these cells are doing in Unit 3. And then the last class of connective tissue is blood. Blood. And there are red blood cells in blood and white blood cells. The white blood cells look purple in histology because their nuclei stain. They're a little bit bigger than red blood cells, and there are different types. So these are the most common white blood cells: neutrophils or polymorphonucleocytes, and lymphocytes are the most common. And then platelets are involved in clotting. And so if you see a blood smear, you will see these little structures, and they are actually platelets. They are valuable. So the textbook that I used to use that these pictures came from has this summary of connective tissues, and it would make sense for you to practice writing out all the connective tissues and thinking, what can I see in that connective tissue? What makes it distinctive? Some of them are going to be easy to identify once you learn them. Some of them are going to be hard. It takes me a long time to figure out what I'm looking at sometimes for some of these tissues. So remember, if you can find identifying features, that's great. Do that first. But if you can't find identifying features when you're trying to identify a tissue, you have to go by process of elimination. So first figure out, is it epithelial? Is it muscle? Is it connective? And it's connective. Can you rule out blood? Can you rule out bone? Can you rule out cartilage? Can you rule out dense CT proper? You have to go like that. So get to know all the tissues as much as you can, and we will practice identifying them in a different video. And I'll see you there.